Good morning, everyone. So I'm Alexandra Hauret. I'm a process engineer, and I've been working for the water department of the city of Lausanne for the past 7 years. During these years, I have the opportunity to participate in the operation of various process pilots, and I've tested innovating measuring devices, including the online flow cytometry monitor, bNovate. And I'm here today to present you these tests and the results that we obtained. To start, the City of Lausanne's water department supplies nearly 230,000 inhabitants in Lausanne and its neighboring municipalities. The water department employs around 200 people, and it ensures the production and distribution of drinking water and the treatment of wastewater. We also monitor the water quality thanks to our internal lab, and we carry out constant maintenance on the water distribution network and our treatment facilities. The water supplies comes from 4 main production plants, which complete the supply of springs located in the north of the city and feeding the reservoirs by gravity. In total, we have 35% of the production from the Saint-Sulpice plant, which has the largest production capacity with 1 cubic meter per second, 33% from Lutry, 16% from Bray, and 15% from Sonzier. The remaining 11% are therefore transported without pumping to the storage tanks. And the water produced passes through 1,200 kilometers of pipe over 500 meters of high differences. So 1,200 kilometers of pipe, 4 plants, around 20 reservoirs, spring, and therefore a need for increasing monitoring, which is why we choose to test the bNovate as a way to improve water quality surveillance. Online flow cytometry tests were carried out with the purpose to evaluate the impact of various steps of drinking water treatment on biological quality, and in particular the stages of filtration and disinfection. We also wanted to check the convergence of the results obtained with the bNovate with those obtained in the lab. This online measurement will allow to reduce the time required between sampling on site and obtaining lab results, and to optimize the quality monitoring of the water produced. Furthermore, the City of Lausanne would like to reduce or even stop the injection of chlorine into its distribution networks, as the City of Zurich already does. So online cytometry would be a good solution to monitor the biological stability of water. Thank you. So we positioned 2 measuring devices, the first one on raw water and the second one on treated water, at 2 drinking water plants, Saint-Sulpice and Lutry plants, and we carried out tests between January and August 2019. At the Saint-Sulpice plant, the devices were positioned on raw water, treated water after sand filtration, and treated water after final disinfection with chlorine. We also operated in parallel of the treatment process an ultrafiltration pilot with or without chlorine process. So we positioned the devices after the UF pilot operated with non-chlorinated backwashing, and after the pilot operated with chlorinated backwashing. And also after the same UF pilot using a non-integral membrane, the same 10 square meters membrane that we used before, and that we slightly damaged. On Lutry, the devices were positioned on raw water after injection of powdered activated carbon, after treated water, after ultrafiltration, which is here operated with chlorinated backwashing, and after treated water after final disinfection with chlorine. So here we can see on this graph all the results obtained during our test campaigns at the Lutry and Saint-Sulpice plants, Saint-Sulpice here in green and Lutry here in gray. The graphs show the number of cells quantified per milliliter, with a measurement step of 200,000 cells. The waters were analyzed every 30 minutes with a Bactosense and every working day by the lab. And at the lab, only total cells were analyzed. On the Bactosense, we use the LDC cartridge for quantification of intact and total cells. So here on the graph, the solid lines correspond to the number of total cells, and the shaded line in a bit darker color to the number of intact cells. For Saint-Sulpice, we see in green raw water with 600,000 to 2 million total cells quantified according to quality variation, of which about 75% are intact cells. In red, water filtered through sand, and in yellow, water filtered through sand, then disinfected with chlorine. In both cases, the number of total cells quantified varies between 500,000 to 1.4 million, with 20 to 30% elimination. The difference is marked here with regard to intact cells. Indeed, after filtration on sand alone, the number of quantified intact cells varies between 400,000 to 1 million, while we see less than 20,000 cells after chlorination. Here in blue, the ultrafiltrated water on the pilot, with or without chlorinated process, and we will focus on it later. Then in gray, the monitoring of raw water at Lutry plant. The values are quite similar to those of Saint-Sulpice, but it's important to remind that both plants take their raw water from Lake Geneva, 10 to 12 kilometers away from each other. And here in purple, ultrafiltration water at Lutry, and we will also focus on it later. And to finish on this graphic, the little circles and triangles of the results obtained by the lab. And we can see the results all well corresponding to those of the Bactosense. Here, this is the same graph, but the measurement step has been reduced from 200,000 to 10,000 cells per milliliter. So we zoom in to visualize the lower part of the previous graph. Data for raw water is no longer showed, and we see here in light orange the water treated on sand filters and disinfected with chlorine for the intact cell part only, as mentioned just before, with less than 20,000 quantified intact cells. Water treated by ultrafiltration for the other compounds shown here. In blue, the treatment pilot in Saint-Sulpice operated with chlorinated or non-chlorinated backwashes and using the non-integral membrane over the last week of tests. In yellow and green, ultrafiltrated water on Lutry, with the chlorinated process, I remind you, and after disinfection with chlorine in purple and pink. Ultrafiltration process gives results of some thousands of total cells quantified and some hundreds of intact cells. And it is noted here that the non-integral membrane allows the passage of a greater number of bacterial cells, below 30,000 total cells quantified. And here too, with the little orange circles, you'll see the results of the lab, which are well corresponding to those of the Bactosense. Here I show you an image of what kind of data the Bactosense has given us for each analysis. This first image is an example of raw water from Saint-Sulpice, with here 500,000 intact cells and 166,000 non-intact cells, and this representation of size and bacterial population. If we look at an image after sun filtration, we have a fairly similar image. And if we now have a look at a picture after disinfection, we observe that the intact cells have been largely eliminated and that there is a greater number of non-intact cells. And on the second and third graph, we observe that the bacterial population size has been reduced, and here it's rather cell debris. So sand filtration has little effect on bacterial removal. This treatment step even provides support for bacterial growth that is necessary for the elimination of some substances. It is therefore imperative to complete the treatment to eliminate the bacterial cells and obtain water suitable for consumption. So the disinfection of water in Saint-Sulpice is carried out by the injection of chlorine. In the case of Lutry this time, the image of raw water is again quite similar to that of Saint-Sulpice. After ultrafiltration this time, both intact and dead cells were removed. Ultrafiltration acts as a barrier and physically eliminates bacterial cells. It is also recognized as a disinfection method with elimination of viruses and bacteria by log 4 and 6 respectively. After disinfection, the image is rather similar to the previous one. The chlorination step did not show any lowering of dead and living cells. However, the role of this step is to limit the resumption of bacterial growth. The quality of the water in the network is then guaranteed by the residual action of chlorine. Here an image of ultrafiltrated water on the pilot in Saint-Sulpice without any use of chlorine backwashing. This image is quite similar to the last two ones. Here we now have a look at the case of the non-integral membrane. We observe that the degraded membrane allows more bacteria to pass than an intact membrane. It is no longer plays its role of physical barrier perfectly. Nevertheless, we observed after some days of filtration that the number of leaking bacteria decreases on the screen after 6 days of filtration. So the hole made in the membrane tended to gradually clog. However, if we were able to clearly observe this phenomenon by degrading a 10 square meters membrane, this would not necessarily be visible in the case of one industrial membrane, which is not integral among several other membranes, especially knowing that an industrial membrane is rather 50 to 100 square meters. And to conclude this presentation, these tests have shown that the cells are not physically retained on sand filters, with 20 to 40% removal of intact and total cells. Living and pathogenic cells are eliminating during the final disinfection under the action of chlorine. This disinfectant, widely used for its persistent action in the distribution network, provided a reduction of intact cells of a factor of 100. The total cells were only slightly eliminated with 20 to 30% elimination. Ultrafiltration behaves like a physical barrier and allows to retain both intact and total cells. With ultrafiltration considered today as a disinfection step, it is possible to reach elimination yields of factor of 1,000 to 10,000 respectively for intact and total cells. Ultrafiltration process allows the quantification of some thousands of total cells and some hundreds of intact cells. Moreover, the results obtained with the online monitoring were in line with the analysis we made in the lab, as a confirmation that Bactosense provides reliable results. And finally, after this pilot test, we made the choice to acquire a Bactosense device. So thank you for your attention.